Hello there, dear students. In today's lab, we will be talking about the non-Mendelian inheritance. Non-Mendelian inheritance, as we said before, that it doesn't follow any law of Mendelian inheritance. According as we all know that we have the Mendelian inheritance and non-Mendelian inheritance. The Mendelian inheritance which follow two important law of segregate and law of independent assortment. In Mendelian inheritance, we know that one trait is dominant over another. But in non-Mendelian inheritance, we don't have dominant or recessivity. For example, if we say that there is a red flower and white flower, the hybrid would be purple or pink. It means red is not dominant over white. The non-Mendelian inheritance is which include the trait of codominance, incomplete dominance, and pleiotropy trait, and others. All of these modes are described in this lecture. First one, we have the incomplete dominance. Incomplete dominance, which occurs when dominant allele is not completely dominant. See, the dominant is not completely dominant. It means we don't have any dominancy or recessivity. The example in this lecture is if we make a cross between red flower and white flower, the hybrid would be pink. And in another example, we have the human hair texture, which shows a incomplete dominance. See, if we look at the figures, the mother is have a straight hair, father is curly hair, and the baby or the offspring, their child, will be a wavy hair. And you already know that how are we going to write the genotype and phenotype. Genotype, which represents the genetic code. In this table, the genotypes are 100% C and S. That is genotype. Phenotype, we are going to describe what we see. Now, at the figure, we see that there's a wavy hair. And CS is a code for wavy hair. The next one, we have the codominance. Codominance occurs when two alleles for a gene are expressed equally in phenotype. For example, we have the blood type. According as we all know that we have four blood types: type A, type B, type AB, and type O. In this type of non-Mendelian inheritance, there would be 6 genotype. In this type, we say that A is dominant over O, B also is dominant over O, and A, B together would be co-dominant. As we said before, the genotype for type A blood, we have AA and I. See, we have an O type. The code for O is a heterozygous for type A. And also with type B, we have IB and IBI. That I is type O. The type AB, we only have one genotype, and type O have one genotype as well. And here is a clear understanding of the ABO inheritance of genetic. As we said, the ABO genes is a autosomal. Where does it locate? The gene is located on chromosome 9. See, chromosome 9 is the locus of ABO gene. What does locus mean? we said before that any gene that occupy a specific location on chromosome, that location is called locus or loci. Here, A and B blood group are dominant over the O blood group. A and B group genes are co-dominant because they are not dominant over each other. A is not dominant over B and B is also not dominant over A. Both traits are appeared in the table, which means they are co-dominant. Each person has two copies of genes coding for the ABO blood group, one from the mother and one from the father. And here's a list of the blood groups, which blood groups can be donated to which blood group. And then we have a example of the codominance trait of non-Mendelian inheritance. See, one parent has a blood group A, and see, the genotype is IAI, which means it's a heterozygous, and the other has blood group B. The genotype is IBI, it's also heterozygous. If we make a cross between these two blood groups, we see that we will be having 25% blood AI and 25% AB, 25% B, 25% O. We will have all the blood group in this family. And then we have the type of codominancy. We have multiple allele traits. What does it mean? Which means it's a trait that controlled by a single gene with more than two alleles are called multiple allele trait. For example, we have a rabbit and 4 color. See color, which gives a full color for the rabbit, which is dominant over other alleles. In this trait or non-Mendelian codominance, we're going to have 4 different alleles. As you can see, we have a C capital and CH, which is a color for chinchilla, and CH Himalga, and C, which lowercase c. C capital is dominant over all other alleles, which means C capital is dominant over C chinchilla and is dominant over C Himalaya and dominant over C lowercase. As well, C chinchilla is dominant over C Himalaya and C chinchilla is dominant over C lowercase, and also C Himalaya is dominant over C lowercase, which means one after another. If we look at the example, let's say that a rabbit was genotype CCH. See, it's a full color. We said before that the capital C is a allele for full color and is dominant over other alleles. Is crossed with a rabbit with genotype C chinchilla C. See, we have the C chinchilla, CCH, and C lowercase. See, in the previous slide, we said that C Himalaya is dominant over C lowercase. So in this case, C chinchilla is dominant over C lowercase. It will be a chinchilla color. What are the possible genotype and phenotype of their offspring? If we make a cross between these two genotype, we're going to find see at the table, it's obvious. Two full color. How are we going to know that there are two full colors? You see, we have the full colors allele at the top, and on the left we have the chinchilla. C capital, which is a allele for full color. It means it comes with any other allele, it would be a full color. Now we have C chinchilla, and C chinchilla with C himalga, C capital with C lowercase, C, and C himalga with C lowercase. There will be two full color because C capital is dominant over all other alleles, and there is one chinchilla and one himalaya. And then we have the pleiotropy. This pleiotropy is one of the most important traits in this lecture. It means some genes affect more than one phenotypic trait, quite the contrary to multiple alleles, the previous slide. They generally involve important proteins that are needed for the normal development. The example for this trait is sickle cell anemia. Sickle cell anemia, which means some red blood cells in human body are sickle shaped and some red blood cells are normal. If we have normal red blood cell and the sickle red blood cell in our body, we would be carrier for the sickle cell anemia, which is a genetic blood disorder caused by a mutation in HBB gene. The normal allele for this trait is HBN, which produce a normal hemoglobin, and HBS, which cause sickle-shaped red blood cell. People who inherit two recessive alleles for this disorder have abnormal red blood cells. If we look at the example, let's say that two parents are carrier for sickle cell anemia. They are carrier, they are not infected, which means their offspring would be infected. The carrier for this trait is HBNS. See, we have normal red blood cell and sickle red blood cell as well. It means that the person is carrier for the disease. If we make a cross between two genotypes, see the father's genotype and mother's genotype, there would be 25% HBNN. It means 25% would be normal and 50% HBNS. It means 50% of their children would be carrier for the disease as well, and 25% have the disorder. They would be infected with sickle cell anemia, and they are going to be sick. Another one, we have epistasis. Epistasis is a circumstances where the expression of one gene is affected by the expression of one or more independent inherited gene. For example, we have two genes. Gene B is dependent on the expression of G1, which means one gene is describing the color and the other gene will decide if the color appears or not. For example, we have BB, B capital for brown, and we have B capital, B small for red. B small for yellow. If the gene that is deciding whether the color appears or not is A small, the color won't appear. It has to be A, or A, lowercase. Both A would be capital and one of them would be lowercase. At that moment, the color will appear. But if both allele are in lowercase, color will not appear. The example for this epistasis we will be doing it during the Venn so that everyone understands it well. I'm going to solve 2 to 3 class, and I'm going to make class between 2 to 3 traits so that you understand it well. The gene whose genotype is expressed, we call it gene A. It's said to be epistatic. This is very important. While the phenotype altered or suppressed, which is gene B, is said to be hypostatic. See, we have hypostatic and epistatic gene. And tomorrow, during the Venn lecture, we're going to solve this problem. You see, we have a brown hair parent and a brown hair parent. Both are brown hair. We're going to make a class between these two traits. Let's see what kind of color we're going to produce. During Venn tomorrow, don't forget. We have the B gene, amount of melange or dark pigment, B dominant, black or brown hair, B recessive, blonde hair. In this case, we have brown hair, blonde hair, and colorless. The other gene that controls the pigment, we have E, which controls pigment expression. E dominant, allow pigment to be expressed. E recessive, blocks pigment, leading to red hair regardless of